All right, let's walk you through some of the slides. Next week is mostly all slides all the time. So, what is that? What organ is this from your quiz? That's a testis. That's seminiferous tubule inside a testis. So, you're supposed to review the testis again. You know this. Let's review now some of the cells. So, what are the cells on the very outside? Spermatogonia, and they're going to make what? Sperm. But you can't say sperm next week because that wouldn't be correct. So if you are between here and here, you're a spermatocyte, which means sperm cell with dollars attached. So spermatogonia or spermatogenic or the outside. Spermatocytes are as you go into the center. When you're in the center in a testis, those are sperm what? Tits. Because they're immature, they become sperm formally at the epididymis. So next week, you can't just say sperm. You got to say spermatogonia, spermatocyte, or spermatid, or sperm, depending on where we're talking. There's a level of detail there. But otherwise, you kind of know this, right? The fun begins when we do the girl parts, because now everything changes.
Can I ask a quick
Yes.
question about that slide?
Sure.
So, was there an obvious substentacular?
That one. That one. I'm going to go with those two. Substentacular Sertoli, substentacular Sertoli. I'll say those two. They just look odd. Usually they're pointy and triangular. Thanks. So the problem is the girl stuff. Let me see if I can find the girl stuff. We're going to use the Boston site here. Okay, so the trick with the ovary is you have to keep track of the stages of the follicle and the stages of the egg, and they don't correspond necessarily. So there was a poster behind this screen. Let me show you the poster quickly. You want to take a picture of this poster with your phone, because this poster will walk you through the steps you're supposed to see on the slides. I'm going to use the poster actually, and then I'll show you the slides. I know it's small to see, but let's go through it. So if you look at your list, way up here on the outside of the ovary, you have little teeny baby follicles that were there before you were born. What do you call those?
Primary.
Primordial, right? Follicles. What do you call the eggs inside a primordial follicle?
Primary.
Primary oocyte. So you have a primary oocyte is in the primordial follicle. So now you have to remember your meiosis to understand what primary and secondary means. Excuse me. What's primary mean in terms of meiosis?
Well, it hasn't even started.
Hasn't been started yet. So that means it's 2N or diploid. What does secondary mean?
Started.
Very good. Started meiosis, so therefore I'm N or haploid. So in a primordial follicle, they're primary oocytes because they have not finished meiosis. They're still 2N or diploid. Does that make sense? All right, so let's do our primary follicles. Our primary follicles look bigger. What kind of oocyte lives in a primary follicle? A primary oocyte, because it has not finished meiosis yet. Let's go to a secondary follicle called the eye of Sauron, right here. So, what kind of oocyte is in the secondary follicle? A primary oocyte, because you haven't finished meiosis yet. Then you got the mature follicle, which is the one that's got the big space on it, also called Graphian or vesicular. What kind of oocyte is in that one? Secondary, because I'm finishing meiosis. I'm now haploid. So on all these lingos, primary means I haven't finished meiosis, or I'm still 2N. Secondary means 1N or haploid. So in the eggs, you have primary and secondary follicles, but they still retain a primary oocyte, because the oocyte hasn't finished meiosis. Make sense? So you can't just go primary, secondary. So primary, secondary follicles with primary oocytes. Here's a mature follicle with secondary oocytes. Make sense? So this primary, secondary has a meaning in this context. If I show you back to my slide, so if I were pointing to these ones on the very outside, which are very, very tiny, and asked you what oocyte is that, you would say primary. If I asked you what follicle that is, you would say primordial. Very good. If I pointed, you'd have to zoom in on a slide, but point to the ones that are a little bigger, more round. Which follicle are those? Primary. And what oocyte are they?
Primary.
Yay. I point to the ones that look like eyeballs. Here and there. There's one down here. The next step up, here's one. They are secondary follicles with
Primary.
Very good. Point to the big circles. Those are going to be mature follicles or grafting or vesicular with. Yay! If you get that, you can beat yourself pretty well.
Can you do it one more time?
Sure. Let me do it on. Let me zoom in. Might be better. Give me just a second. So we're zooming in closely on an ovary. Here's a little, this would be really, really tiny, with not much structure. So that follicle is primordial. That egg is, yay. Too easy, drill sergeant. Let's try this. Okay, the one at the top is what you just named. The one at the bottom. What is this one? The follicle-wise? Primary, because you can see the circle. And this egg is the primary, because it's not done meiosis yet. So I'm gaining in the follicle and growing the egg. So try this one. This is not the best one in town. This one actually should have a space on both sides, but it has only one. So just pretend with me that looks the right way. So what kind of follicle would be the next step? And what kind of oocyte? Primary.
So
Very
you look
good.
for space around the periphery?
Yes. So you're gaining peripheral space, and you're gaining size. So then our last one, my favorite, the one that's full of fluid with the egg on the side. So this follicle is mature grafting vesicle. That egg is secondary. Here's some little primordials over here. Right. So you're going to have to name the cells and the egg separately in your head as you go up the chain. Make sense?
Is there a picture that has all of them compared to each other?
On the poster.
Okay.
So the poster is going to be your friend. So let me do, if I could get Boston to work for some reason, you can zoom in, but it doesn't want to zoom. I can't get the zoom to work anymore, but they used to have a really nice one if you go right here. Primordial, primary, secondary, vesicular, all in one little scan. But it won't zoom in anymore. Damn them. All right. Let's find a uterus. That's where life gets fun. Okay, for the uterus, it's the same slide you saw last week. But now you have to know what stage of your menstrual cycle it is. So what you're going to do with your head and your eyes and your brain is you're going to look at the uterine glands. And you're going to compare them between the different slides. So looking at the uterine glands, these little worm-like things, describe them to me. What shape are they in? They're kind of straight. There's a few that might be bent, but they're mostly straight and or round. This is when the uterus is growing in thickness. So when do you grow a uterus in thickness? The follicular phase. But in a uterus, we call it the proliferative phase because you're growing, you're proliferating. So although it's the follicular phase of menstrual cycle, it's the proliferative phase of the uterus. Right? So that's your proliferative phase. They look like they're long and straight and not too bad. Okay, compare what you saw in your head to this picture. Tell me about the glands. There's more of them. What else do you notice about their shapes? They're convoluted, like that word. It's very good. They're bent, corkscrewed. There are very few of them are straight or round. They're a lot more squiggly. So this would be after you've ovulated as you're making it thicker and more bloody. So what phase is that in uterine terms? Secretory, because you're secreting more stuff. So proliferative is before ovulation, secretory is after. That's secretory phase. The worms, we call them, are getting more crooked. Okay. So then, let me show you this picture here. This is from far away, but you can see the uterine glands up there. So what phase do you think that is? If they're straight and long.
Proliferative.
Proliferative, you're growing. So proliferative phase, straight and long. On the left-hand side, you'll have to probably squint to see it, but take a look at there and tell me what you think that phase is.
Secondary.
That's secretory. There's a lot of them. You can see they're all different shapes. They're kind of bent. So that would be secretory phase after ovulation. So you can see the number changes and their shape. This one should be self-obvious. Menstrual phase. What happens in menstruation? That looks like your uterus is coming off, because it is. So they're breaking off. You can see dead cells. If you zoomed in, there'd be blood cells in there. So they're basically busting the worms apart. Right? Yes, takes all the romance right out of it. Right? Those are the three main phases. So what I'm going to do, I'm going to randomly go through and find a few phases to scare you with. Let's try this one. Let's try this one. Okay, give me a phase. How many uterine glands do you see? Not that many. What shape are they? Kind of straight. Yeah, this one's crooked, but most seem kind of straight. Therefore, this is proliferative. There's very few glands. They're mostly straight and round. So, before ovulation. Okay, Smarties. Let's hit you with the hard one now. Name it and claim it.
Secretary.
Secretory. How'd you know? Lots of glands, squiggly. Notice they look jagged. You know, corkscrew. Very good. Yes, I know it. Give me just a second. Let me do this one here. What organ is this? It's an ovary. How'd you know? Look at all those follicles, right? So now we're going to try doing our follicles. If I were pointing up here to these little tiny circles, what do you think that would be? Primordial. What oocyte do you think that is? Very good. If I point to this one, give me its follicle. Mature. Give me its oocyte. Too easy, drill sergeant. How about, let's do this one right here. Or here. Here's one.
Secondary.
Okay, I'll give you secondary. Secondary what, though? Follicle or oocyte? Follicle. So the oocyte must be... Yay.
What's that big one on the right?
This? That one was one of these that already or is going to ovulate, so they just missed the egg when they sliced it. So they just kept... That would be the antrum of the mature. They get kind of...
You said that you'd accept secondary, what were you going to say?
I'll say that's a late primary, personally. But that's me. Let's zoom in. To me, that's a late primary. I've done it before that. Okay. The thing on the left is what I'm interested in, not the things on the right. Corpus luteum. How did you know that was a corpus luteum? It looks like one. Indeed. Corpus luteum, they look puffy, cloud-like. They don't look like round follicles. That's the old follicle. When this egg left, or will leave, they'll get this. So there's a corpus luteum. They look puffy, cloud-like, some people say. Corpus lutea there. So then we're going to do... in the upper left, compared to the right. Albicans. How'd you know that was an albicans? It looks like one. Yeah, they're shrinking, right? So the luteum gets smaller and smaller and decays and becomes an albicans. They look kind of the same, just size is different. This one looks a little more destroyed. But
So that large one on the right is trying to become an albicans.
yeah, that's a luteum going to become an albicans. Right. So I've got a little version here. Here's another albicans. Looks like a puffy cloud, but notice how small it is compared to the follicles now. So these are follicles. So that's just shriveling up and becoming a scar. So the luteum is usually big, the albicans are usually small. So just I don't want that one. look at the size again on that one. The luteum's bigger than all the follicles, right? It's huge. And the albicans would be smaller than the follicles.
Can you? Where's the albican on here? Just
There's no albicans
Oh,
in this one. There's all luteum.
okay.
That's just one big one?
Yep, that's one big luteum. So it gets bigger than the follicles it has. Can anyone see a corpus luteum in this picture? That's the whole thing you're seeing here. Well, to show you that's during pregnancy. Right? So if you think about it, remember we learned that in lecture that you have to maintain the corpus luteum for a couple weeks. So when you're pregnant, it just gets about like half your ovaries sitting here as a corpus luteum. Then eventually that will then shrink down. Right? But so again, a corpus luteum looks like a big cloud. Right? Make sense?
And they don't shrink down to the same size as any other.
Yeah, they'll eventually shrink to any other.
And is that once implantation is taking place?
Pretty much, yes. So
Is that
once ACG basically drops off
Right.
after that. Right. Is it okay? So you want to spend time getting your ovaries down. That's the majority of the slides will be the ovary stages and the uterine stages, like all of the poster. But you're also supposed to remember your meiosis processes from 112. Does everyone remember meiosis from 112?
No.
Okay, 'cause you're supposed to remember that. P-phase 1, metaphase 1, blah, blah, blah. So, I'm not going to necessarily go through it again, but let's watch a small animation about meiosis. Please remember how it happens.
This is the process that results in the formation of sperm cells and egg cells. The cells that will undergo meiosis are typically found in the testes and ovaries of males and females, respectively. These germline cells are diploid, having 2 sets of chromosomes, but will undergo meiosis to produce haploid cells, having 1 set of chromosomes.
So how many chromosomes are in the diploid set of a human being? 46. How many are in a haploid set? 23. So for 2N and N, you could substitute 46 and 23 for a person. For other animals, it's different. Or you could say 2N and N, or diploid and haploid. Remember that language from 112? Vaguely? Yeah. Okay.
During fertilization, these haploid cells fuse to form a diploid offspring.
That's the zygote.
Like mitosis, meiosis is preceded by an interphase. During this time, DNA replicates and each chromosome becomes doubled, consisting of 2 identical strands of DNA.
Or the chromatids.
Meiosis involves 2 divisions. The first division, meiosis 1, halves the number of chromosomes, and in the second division, meiosis 2, the sister chromatids are split. The end result is 4 haploid cells.
Every time. Always 4.
Now let's look at each of these divisions in more detail. Meiosis 1 begins with prophase 1. Individual, replicated chromosomes become visible, and each chromosome consists of identical sister chromatids joined at their centromeres.
Remember that?
The spindle fibers start to form, and the nuclear envelope disintegrates. In addition, homologous chromosomes line up next to each other, and an event unique to meiosis occurs.
What is that unique to meiosis event occurring? Yeah, it's a crossing over. You can say synapsis, which is another way to say it. Chiasma is another word you'll hear, but basically homologous chromosomes pair. Remember, homology meant you're the same. So dad's chromosome in purple and mom's chromosome in green pair up and begin to swap places. And that happens in prophase 1 of meiosis. That's a unique feature. That's homology, or homologous chromosomes pair. Also known as a tetrad. So when your lab talks about tetrads, you count the chromatids. 1, 2, 3, 4. Tetra. So the 4 chromatids are what occur in the crossing over. There are 4 of them there. Also called a bivalent in the Dark Ages, which are 2 sets.
During this unique event, called crossing over, 2 homologous chromosomes, which are lined up next to each other, exchange DNA between adjacent homologous chromatids. After crossing over has occurred, the sister chromatids of one chromosome are no longer identical to one another. In metaphase 1, homologous chromosomes line up along the equator of the cell. For each pair of homologs, the orientation on the equator is random. Currently, the large purple chromosome and the small green chromosome are on top. However, the small purple chromosome could just have easily have ended on top. Each different orientation results in gametes with different combinations of parental chromosomes. This process is called independent
Does that ring
assortment.
a bell from Mendel?
Once the chromosomes are lined up, the ends of the spindle fibers attach to each centromere.
What comes after metaphase?
In anaphase 1, the chromosomes move apart from one another along the spindle fiber to the opposite ends of the cell. At this point, each chromosome is still double-stranded and has two sister chromatids. However, we are separating homologous chromosomes into two different cells, so that each new cell will have only one member of the homologous pair and will be haploid. In telophase 1, the spindle fiber disintegrates and cytokinesis, cell division, begins. Thus, in meiosis 1, which progresses from prophase 1 to telophase 1, two cells are created, and each cell has half the number of chromosomes compared to the original cell.
Therefore, you make two cells that are haploid, right? But you're not done, you got to split again. That's where you get the 4 cells. But you're going to split just the chromosomes apart, not... You're going to basically split the chromosomes apart from their chromatids. So you're just going to double that, basically, yes.
So, like, when it's all said and done and you have the whole bodies, are those also haploid
Yes.
cells?
Yeah, so if this were a female, one of these would stay an egg, let's say this one. This one now is your first polar body, because it doesn't count. Then this one would split again, and then you'd have another polar body. If this were a male, both these would go on split again, and each one would make 2 sperm. So, but the polar bodies is because you have to make the 2 splits, and so one of those would randomly shut off. So let's do that. Go with that.
After cytokinesis is complete, a second division, or meiosis 2, starts. This division is identical to mitosis, and the resulting cells have the exact same number of chromosomes as the original cell at the end of meiosis 1. In prophase 2, we now have 2 cells, each with 2 chromosomes. In this stage, the spindle fibers again start to form at the poles of the cell. In metaphase 2, the chromosomes line up along the equator. This is different from metaphase 1, where the homologous chromosomes were lined up. In metaphase 2, each cell has only one of each homologous chromosome. In anaphase 2, the sister chromatids move away from each other along the spindle fiber, and in telophase 2, along with cytokinesis, we see the formation of 4 genetically different haploid cells.
Make sense? Make sure you can run your mind back through meiosis. There's some language in the lab manual you're supposed to know. There's also a picture you should know that if I just gave it to you on a quiz, hint, hint, that you can label haploid, diploid, prophase 1, metaphase 1. Go back to 112 and reproduce the 2 weeks you spent learning this and forgotten by now. All right? Make sense? So you got to know your meiosis words. You got to know ovaries and testes. That's pretty much the lab. So it's pretty light in terms of what you have to know. It'll be mostly paper pencil. Make sense?
Is there any website that you think is